So welcome everyone to the 160 or so people who registered to attend today. I hope that everyone did get to dial in. This will be a bit different from other KHI webinars we've had. We'll kick it off with some brief comments from three speakers who really need no introduction. But the majority of our time will really be geared towards listening to you all. We really want to hear your perspectives, the good, the bad, and the ugly on data sharing. IgA nephropathy was first described in 1968, and it took 53 years to achieve the first drug approval. I dug up our KHI proposal, the original one back in 2015. Thank you, Dr. Nachman, who I see is on the line. And back then, there was really only one question we were trying to address: which outcome measure could serve as a reasonably likely surrogate endpoint and thereby open the accelerated approval pathway. Data sharing provided us with the analyses we needed to identify proteinuria as that reasonably likely surrogate, which also facilitated 2 drug approvals and an explosion of IgA nephropathy trials. Nearly 180 were on clinical trials as of last summer. So with so much new data emerging from these current trials, we now have the liberty to pose more questions. And the answers, again, will be found within the treasure troves of accumulating data that we're seeing today. That, again, needs to be shared to facilitate more meaningful analyses, which brings us to today. Abdul?
Yeah, great. Thanks, Barb. I also want to just add my warm welcome to you and look forward to a really robust discussion. As Barb mentioned, we're honored to have a distinguished panel of internationally renowned speakers who are going to share their insights on this important topic. So first, we'll have Eliza Thompson, who serves as the Deputy Director of the Division of Cardiology and Nephrology, Center for Drug Evaluation and Research at the US FDA. She'll delve into the significance of proteinuria as a surrogate endpoint in IgA and explore the journey of data sharing that has led us to this exciting point, as well as provide valuable insights into the current FDA perspective on clinical development for IgA. And then next, we'll have Jonathan Barrett, the Mayer Professor of Renal Medicine at the University of Leicester, who'll shed light on the unmet needs in IgA that remain and lay the groundwork for addressing future questions. So he'll discuss ongoing studies that may support alternate surrogate endpoints and outline what requirements might help establish proteinuria as a validated surrogate endpoint. And then last but not least, we're lucky to also have Guido Lambers-Heerspink, Professor of Clinical Trials and Personalized Medicine at the Department of Clinical Pharmacology at the University of Medical Center, Groningen, who will share his expertise on data sharing and provide examples of successful practices in this area while offering valuable insights into how we can effectively navigate the challenges of data sharing to advance research and ongoing development of therapies for IgA. And then finally, we have all of you. And as we transition to the listening session, we really encourage you to actively participate by considering the barriers to data sharing in IgA, help us explore potential solutions, consider realistic timelines for new initiatives, draw lessons from within, inside and outside the kidney space, discuss patient concerns regarding data sharing, and examine ways we can maximize industry support for these efforts. So with that, I'll turn it over to Eliza to kick things off.
Great. Thanks so much, Barbara and Abdul. And like Abdul, I just want to thank you all for taking the time out of your very busy schedules to participate in this session today, which is really intended to be a listening session, at least for us, at least for me. I think it's fair to say, and certainly I think both Barbara and Abdul noted this, that data sharing transformed the drug development landscape for IgA nephropathy. How? I think on the one hand, data sharing very much gave us a better understanding of the quantitative relationship between early changes in proteinuria and later effects on loss of kidney function and progression to kidney failure in patients with IgA nephropathy. At the same time, data sharing efforts led by people such as Guido gave us a better understanding of how well changes in the rate of loss of kidney function assessed over maybe just a few years predicted treatment effects on progression to kidney failure in a much broader population of patients with chronic kidney disease. And I think it was really both of these data sharing efforts, both of these efforts were needed to really understand how to design trials that could support accelerated approval in IgA nephropathy, as well as verify the benefit of these new treatments. So, that's really how we got here today, right, with data sharing. But what about the future? I think a lot of important questions are coming up. Certainly we're being asked questions, and presumably you're asking some of these same questions. Could we use early changes in proteinuria alone, or possibly in combination with early effects on GFR, perhaps at 1 year, to determine the efficacy of new treatments for IgA nephropathy and perhaps support traditional approval? Should we be using a different approach here to evaluate the efficacy of our treatments, either when used alone or in combination with other treatments for IgA nephropathy? And I think also other questions might be, could we do a better job of identifying who is actually at high risk of progression and who is not? I think if we share data, we as a community can answer these questions as well as many, many, many more. So with that, I am going to stop talking and just again, thank you all for participating today in this discussion. And again, just want to thank you for your willingness to share your perspectives. I do very much hope you'll speak up. I'll turn it over, I think, to, is it John who is next?
Hi, I'm John Barrett from the UK, and I've been asked to help set the scene for the discussion today. I've been asked to address two questions today. The first is it time to reconsider the approval paradigm in IgA nephropathy? And to do that, I'm just going to remind everyone what the unmet need is for patients living with IgA nephropathy. So this is data based on the UK's Rare Disease Registry, RaDaR, and was published last year. It's the largest global registry of IgA nephropathy patients, and I'm just going to show you the outcomes with current available treatments. So if we look at this data, if you're a child diagnosed with IgA nephropathy, 20 years after diagnosis, half of those children have developed kidney failure, which means they develop kidney failure in their 30s. If you're an adult diagnosed with IgA nephropathy after 20 years, 3/4 of those adults have developed kidney failure, and that's kidney failure in their 50s on average. And this data isn't peculiar to the UK. We've had data presented at the ASN last year showing very similar data from the United States and from Canada. What we look at here is based on the GFR at presentation, what rate of loss of kidney function do we need to achieve for the rest of the patient's life to avoid kidney failure in their lifetime? And just as an example, if we take people diagnosed with IgA nephropathy between the ages of 18 and 30, using their real GFR at time of diagnosis, you can see the likelihood of developing kidney failure depending on the rate of loss of kidney function for the rest of their life. And you can see we only make a significant impact on the risk of kidney failure when we get down to rates of loss of kidney function of 1 ml per minute per year. And actually, when you look across the age groups, you can see that's entirely consistent. And so to prevent kidney failure in the lifetime of our young patients with IgA nephropathy, we need to achieve a rate of loss of kidney function of 1 ml per minute per year for the rest of their life or lower. And unfortunately, with available treatments and those treatments that have only recently been approved, we are getting nowhere near this level of kidney function loss. And so there is a significant unmet need for new therapies or combinations of therapies that are going to achieve this slowing of kidney function to the point that we can be confident our patients will not experience kidney failure in their lifetime. Now, we know that proteinuria is a very good predictor of how quickly a patient will develop kidney failure, not whether they're going to. I've just shown you data telling us that they're going to develop kidney failure, but how quickly. What the RaDaR data tells us is that even at low levels of proteinuria, so less than a gram, less than half a gram, as you can see here with two different ways of measuring that at those levels of proteinuria at 10 years, 1 in 4 IgA nephropathy patients develop kidney failure. So we need to start rethinking the thresholds potentially for going into clinical trials, but also what our goal is in terms of proteinuria reduction in the short term in protecting against lifetime risk of kidney failure. So I'll just remind you of all the work that went into establishing proteinuria as a reasonably likely surrogate of future kidney failure. And that was a significant amount of work driven by a global collaborative approach with sharing of data, very robust statistical analyses by Leslie Inker and the group at Tufts, and a collaborative approach within the KHI between the regulators, between academics, between industry. And this is where I hope we will move to when we reassess where we might want to be in the future in terms of endpoints for trials in IgA nephropathy. So if we think about IgA nephropathy, we have those basic pathophysiological processes at the far left of the picture, moving through to changes in proteinuria that are measurable and, as I've said, are an approvable endpoint, moving on to changes in kidney function or slope of GFR, again now recognised as an approvable endpoint, and finally to dialysis. We have full approval based on GFR changes over 2 years, and we have an accelerated approval based on proteinuria changes. So what is it going to take to change this paradigm? And that's the second question I've been tasked to discuss today. I'm just going to give some thoughts as to where we could go if we're able to generate the data and able to identify with clarity that these changes in endpoints are reliable and robust. So could we move GFR changes to a 1-year time point? That would certainly shorten trials, could be potentially used for full approval. That's something we should discuss, I think, in the meeting. We now have 2 trials, phase 3 trials, that have reported proteinuria changes and GFR changes, and there will be more coming. Will this deliver data that could allow us to consider a change in proteinuria as an endpoint for full approval? Does proteinuria change from a reasonably likely surrogate to a true surrogate for end-stage kidney disease events? Again, something we should discuss later. Could we in fact actually think about a combination endpoint, combining proteinuria and GFR changes? That's certainly being considered, and we've got Hiddu here who authored this paper. I'm sure we'll discuss this later. Could this be an option? And then we now have trials showing that actually we can impact on the fundamental pathophysiology of the disease, particularly measuring Gd-IgA1 and circulating immune complexes. How might we approach that and think about whether these changes could be used for an accelerated approval? What data would we need to be able to look at that? And so I come back to my second question, which is, how might we change the current paradigm for endpoints in IgA nephropathy? And I think it's important for everyone here today to understand that we are only going to be able to consider these potentially new endpoints if we have access to high quality clinical data that will allow us to perform those robust analyses. And this is where Hiddu is going to take over and talk to you about how we might get to that point of data sharing.
Dear colleagues, it is my pleasure to give you an update about our data sharing experience within the CKD-EPI Consortium. The CKD-EPI Consortium was initiated in 2003 with support from the NIDDK to address fundamental questions in CKD epidemiology in order to create useful tools for research, patient care, and public health. The consortium is led by Leslie Inker, Tom Green, and myself. And the coordinating center is located at Tufts Medical School in Boston, and Leslie Inker is the main coordinator. Validation of proteinuria and GFR change as surrogate endpoints is a major objective of CKD-EPI. CKD-EPI held milestone scientific workshops in collaboration with the FDA and EMA in 2008, 2012, and 2018. In order to validate proteinuria and GFR change as surrogate outcomes, one needs a very large database that includes different disease subclasses and different interventions. We managed to develop such a large heterogeneous database that now includes 66 randomized controlled clinical trials in patients with different types of chronic kidney disease that also includes different interventions, as can be seen in the table on this slide. To validate proteinuria and GFR change as surrogate outcomes, access to individual patient data from these well-powered RCTs is critical. With the support from many collaborators, we managed to obtain access to the individual patient data through different ways of data sharing. Either data transfer agreements for direct transfer of data were established, or we shared data through a central server, for example, the FIFLI server. During the years, we have experienced different and multiple challenges. And these challenges can be classified into different categories as shown in this table. First of all, we saw challenges with respect to the data acquisition, and in particular with respect to the study identification. It turns out that there are different data sharing platforms, and they are not always easy to identify. To solve this, we believe that we should increase the transparency of data sharing platforms, and I will abbreviate this as DSPs, by integration of these different platforms. We've also seen challenges with respect to data agreements, in particular the approval and the duration to obtain approval. Initially, sponsors are concerned of not adhering to privacy laws and regulations. In addition, it takes a long time to set up an agreement, typically between 3 to 12 months. We believe that the data from RCTs should become available as soon as the regulatory authorities have approved the drug in certain markets. That's not always the case nowadays. And in order to speed up the process of the agreement negotiation, we believe that standardized agreements could help with pre-specified choices so that we don't have to start every time from the beginning. But if we would have a standardized agreement, that would smooth the negotiation process. There are also challenges with respect to data analysis. It turns out that analyzing data through DSPs is difficult because of the computation speed and the insufficient storage that is often at these DSPs. So we have to improve the interoperability of DSPs to increase computing resources. But quality control is another problem. Often there's inadequate information on the different variables that are stored in a database. Clinical data interchange standards are needed to provide a common terminology, which would at least partly help to solve this problem. And then finally, we've seen issues with the dissemination of results. Sometimes we were unable to address questions from editors and reviewers because the contract that we had with a collaborator had expired, and we were not able to address questions from the editors. So the agreements that we have with our collaborators should only terminate at the completion of the research when the main manuscripts have been published in scientific journals. So the lessons we have learned is that every database has its own challenges. Before analyzing data, examine the database by plotting histograms, replicating the characteristics, in particular the baseline table, as well as the outcomes table. We also believe that we should establish the point person from the sponsor who can help to explain the database and can help with the data analysis. And then finally, working closely with the contract office to explain the nature of the agreement is very important. We have recently published our experience in an editorial that was published in JSON at the end of last year, and I recommend everyone who is interested in this topic to read this editorial. Then finally, and most importantly, I would like to acknowledge the tremendous support and contribution of all the collaborators, because without their support, we were unable to conduct our research, and we always do this together with our collaborators. I thank you for your attention.
So Hidde, would you like to kick us off on this discussion?
Yeah, well, I think this was a fantastic presentation by John Barrett, and I believe that the future may lie into the combination of these surrogates, albuminuria and GFR slope, and think about how can we best combine the two to have a good prediction of what a drug effect will be in IgA nephropathy going forward, which potentially could even lead to a direct full approval. The paper that we wrote sort of tried to combine the best of the two worlds, GFR slope and albuminuria. Albuminuria has the advantage of being able to show an effect early on, although the association with the clinical endpoint is a little weaker than GFR slope. But to demonstrate a treatment effect on GFR slope, you need a longer follow-up. But if you could combine the two, you could perhaps have a shorter follow-up than GFR slope alone, and but still have a very robust indicator of the treatment effect on the clinical endpoint. So maybe that is one way for going forward, because I believe that with all these improvements in therapy, and if we start thinking about combination therapy, and if the rate of progression is already decreased in the background population, the trials will only become larger. But I still believe that it is necessary to think about the future, think about the sequencing of the drugs and which drugs to start with, and also thinking about personalization of therapy, which drug or which combination of drugs are best for each patient. So these are some of my ideas: personalized medicine, sequencing of drugs, and the ability of developing new endpoints.
Thanks, Hidde. John, I wanted to give you a chance to say hello to everyone as well, and see if you have other questions you want to pose.
Yeah, thanks, Barbara. So I think one of the things that's come up in discussions is with new therapies, how do we approach combination therapies, or how do we approach a trial where we are not using a placebo but an active comparator? For instance, two drugs that target the same physiological mechanism. And what would that trial look like in a rare kidney disease? In terms of, does it have to be a non-inferiority trial? How might we look at that, where we're now having drugs that are coming in that are targeting similar mechanisms and may well be approved? So I think that's going to be something I think that's really worth thinking about, because we don't have that barren landscape that we had 5 years ago, thank goodness. We have new therapies coming, but as I hope I got across, we're setting ourselves a really high bar to prevent kidney failure in the lifetime of our patients. You saw the rate of loss of kidney function we are going to need to achieve in our young people who are presenting late. And I think combination therapies and combination approaches are going to be critical to that. And it would be good to rehearse the arguments of how we assess those in clinical trials of a rare kidney disease, which may not be so straightforward compared to trials in perhaps DKD or other conditions of general CKD.
So lots of good questions about trial design. I see some questions in the chat about kind of treatment duration and other things, but I want to make sure we really focus on our objective here, which is the listening session and hearing the perspective from industry and people in academia who share data and registries. Tell us what you think the challenges and potential solutions are. We had a nice slide from Hidde that started to kick off that conversation. And I see a lot of names in the chat who I know have experience with this and have thoughts about this. So
while we're sorting that out, would it be okay if I just asked Elisa a question?
Oh, absolutely. Oh, and
Yeah.
I forgot to give her a chance to speak. Go ahead.
I just think it's worth setting expectations about how important data sharing is. And that is, what would it take for any of those endpoints I hypothesize could potentially be useful? What is it going to take to be able to get any of those approved by the regulators? Because I think we need to understand it's a really good aspiration, but we are really going to need to do some hard work to convince regulators that they are something that could be used to approve a drug. And I think it's worth just setting the scene, Elisa, in terms of what we've got ahead of us to be able to get to any point that's different to where we are now.
You know, I actually, though I will say that the bar is high, to accept something as a validated surrogate endpoint. I think we're actually in the trials that are ongoing are going to be able to help us answer this question. And so I actually feel quite good about the ability to come up with a better way of doing this. I think it'll take obviously discussions. I do want to flag, though, that I think the goal of today was really to hear from our participants.
I think I'm going to go ahead and call on Alex Mercer. Alex has worked with many of us across different projects. We're currently involved in the Paracel project, which similarly, we're trying to get folks to share data so we can look at potential surrogate endpoint for FSGS. So Alex, I know you've got some thoughts on data sharing, some of the challenges from the perspective of industry.
Okay, so I think we have our first wave of therapies in IgA coming. Some have been approved, and we hope that there's going to be a series of more different mechanisms of actions which are going to be approved. And this is going to revolution the landscape. It will slow down the rate of loss of kidney function, which also makes challenges for the next wave of therapies to come along, because the rate of loss of EGFR is going to be less, and it's going to be harder to show a treatment benefit. So I agree, data sharing has to take place to enable proteinuria. We hope the data supports to become a validated surrogate endpoint. But we also need to think that there are multiple stakeholders in here, and I'm putting an industry hat on here. And I think it's the timing of data sharing needs to be thought through for industry. So a lot of those companies have taken great risks and enormous sums of money to develop drugs and be the pioneers in IgA nephropathy. And to share data immediately after approval, as you were mentioning, comes with challenges in that it's enabling the next wave to come, and it impacts on the commercial opportunities to recoup the money which they've invested in. So I think we definitely need to think about when this can take place reasonably, when you can expect sharing of data. We can also think maybe for SGLT2 inhibitors, which are about to go generic, that's a different circumstance for an enterprise. And so maybe for those companies, it's far easier to share data when the drug has been available in multiple indications for a very long time, and it becomes a generic, compared to companies who have a recently approved drug, and maybe this is their main source of revenue, and to recoup their return on investment. So I think it's the only way, data sharing is the only way we're going to move this forward. But there is that element of timing, and we need to put our feet in, from an industry perspective and what their stakeholders need. These are commercial groups. The initial data sharing for IgA nephropathy, if we look, and it was kindly from investigators and investigator-initiated studies. So I think this perspective needs to be considered, but I think we're all in agreement for the best of patients that we share data as soon as possible, but there is this reality as well. I'll stop there.
And Alex, this is Hedo speaking. From your perspective, and if you put your industry hat on, what is a reasonable timing? I mentioned after drug approval. Do you consider that a reasonable timing? Or does it depend on the size of the company? Does it depend on the patent life? And having all of this said, still one benefits. Even companies that contribute data also benefit by contributing data, because there's new science, new data can also be used by the company.
Absolutely. All of those things come into play. And each company is going to have their unique set of circumstances which they're in. So I think we have to appreciate that. They've benefited the sharing of data, but also think from their perspective. If in 2 years' time, proteinuria is a validated surrogate, new drugs will come on the market very quickly, which would reduce the value of their product. And of course, that is how palatable is that to shareholders? That's a challenge. So this is coming from, people are going to think I'm nasty now, but this is I think the reality which we need to consider. I'm not speaking for any company here. I'm an independent consultant, but it's just I'm trying to put on my hat as if I may be them from their perspective. So I think each company will be different.
Understand.
Prabir, I see your questions. Would you like to go ahead and verbalize them?
So first of all, yeah, this was some great initial presentation. So it was really a two-part question. The first was to get the FDA's thoughts about their interest or positiveness or otherwise about having biomarkers, if you will, as part of the composite endpoint that Hido spoke about. And then the second part that's linked to it is that, and this is, I guess, a question to industry. We've spoken so much, obviously, about sharing of data, and Hido, you've had a really successful example, but that's sort of been data of eGFR and proteinuria. What are industry's thoughts about a pre-competitive data sharing that also involves samples? Because that would feed into the first question about biomarkers, even if the samples are just from control groups, let's say in RCTs. And I know that's much more complex, but wouldn't that be wonderful if we could have that? Because now we're not just identifying new biomarkers for surrogate endpoints, but it would allow us to really identify druggable targets, go into pathogenetic pathways, and there is so much data that we're collecting in terms of samples, and so much. And we've moved so much ahead in ways of analyzing that sample data, both with regard to technologies and analytics. I will stop, Barb.
And I think, Prabir, you may have asked me a question, and so maybe I'll try to quickly answer it, and then we'll hopefully hear from industry. Obviously, things like proteinuria and GFR, these are all biomarkers, right? Patients don't complain of these things. Their significance really lies in the ability of treatment effects on these things to translate to treatment effects on outcomes. And so I think, yes, I think there is interest, and even you're seeing in the chat about exploring other parameters, other things we measure in the blood or the urine or on biopsy to predict outcomes. But really, the only way we're going to get there is if we find incentives and make it attractive to share data. Again, I think it gets to this whole issue of the reason we were able to transform this space was because of data sharing. And I think the extent to which we're going to be able to really ensure that we have a meaningful impact on the lives of people who live with IgA nephropathy and then have a meaningful impact on their risk of progression to kidney failure. We're going to need to find a way to come together to create incentives to make it attractive for industry to share the data. I do want to just add that I think there needs to be some thought to what data we actually have from these trials, right? And what's going to be feasible in the long term versus what's going to be feasible later. But I'll stop, and hopefully we can hear from industry as well as patients and others in terms of how we can make this more attractive, what is feasible in the short term, what's realistic to expect. And I just want to say thank you to Alex for speaking freely and speaking to some of the challenges we're facing.
This is Julie Lynn from Sanofi. Prabir, to answer your question, I think your vision is exactly the right one. It goes far beyond kidney disease. As someone who's worked primarily in a variety of rare diseases, this idea of many stakeholders pooling data, clinical data, as well as ideally biological samples would be definitely very helpful. However, I do want to point out that there's a lot of work that's involved in preparing and anonymizing data sets, and then also then combining them into one data set that can be analyzed. So in terms of sponsors and in terms of academic centers and other institutions or parties that have those data that could be pooled, where are the resources going to come from? And what are the incentives, as Aliza has pointed out?
Yeah, thank you both for those answers. I'd love for Julie, for other people to speak about the two things you raised, which I think are critically important, i.e., incentives and potentially regulatory agencies maybe could be a part of that, and then the other point that you mentioned, of course, about just the sharing pre-competitively.
Can you guys hear me?
Yes. Hi, Whitney.
Okay, perfect. Hi. So definitely the data sharing would be so helpful, not only just from patient perspective, but I know definitely from y'all's perspective too, because trying to get things over the years that you need to gather all the information for these studies sometimes is a little bit harder. Better now since most people have the computerized systems, but to gather all of that, personally, whenever I went to be, look into a clinical trial, I had an entire little binder for them, but it's just not something that everybody has or has access to. So definitely beneficial for the patient if they don't want to try to search for all that, but also you guys. Yeah, it's definitely a big hurdle because I know data sharing, there's always the risk of breaches and things to that nature, but that would be so amazing.
Whitney, can I just ask, it's John Barrett here. As a patient, and I think you've been in a clinical trial or you certainly talk to people, do you feel patients would be uncomfortable with their data being shared in this regard?
I don't think they would. As long as, so the big thing about that is they don't want it to be shared with, say, a pharmaceutical company and it be used without their knowledge. So many of them are very, personal about that, where, listen, your information is only going here, for continuity of care and all those things. So that's just being upfront with them, what the information is for, and who has access to it is really a big thing. And a lot more patients would be open to it.
So this is a great conversation because I see a comment from Kevin Fowler in the chat, and I know Curtis is on the line too. It feels like we have not done enough, if anything at all, to really educate and make patients aware of, the why, the how, and how we protect their data when we talk about data sharing. So if that's one action item we need to take off as a collective group, it doesn't matter who picks this up, but that's a really important topic. And maybe at some point we have a panel that's the voice of the patients to really understand some of the intricacies involved and the levels of discomfort and how we can kind of address and alleviate those concerns.
Because definitely if there's one thing that we've kind of pushed with all of these IgA patients is be your best advocate, ask all the questions, know what's going on. And sometimes that plays beneficial and sometimes it kind of nips us in other areas, but very much leaving people in the dark, not meaning to, of like where their information is going or what's being shared. That's a perfect thing as to why up to them being more open to it if they have that information up front. So, yep.
Sure. Okay. Thank you, Whitney.
Hey, can everybody hear me? Hi, Barb. How are you?
Hey, good. How are you? We can hear you.
Good. Terrific discussion. Jonathan's wonderful as always. And I want to go back to something Eliza made a comment about sort of the evolving biomarkers, urine and blood. And many of you know that I've always been a strong advocate for biopsies. And I think the point that I'd like to make is I'm sure many of y'all saw what I thought was a fantastic abstract that Heather Reich had at last year's ASN meeting, where she demonstrated the presence of gal-deficient IgA1 plasma cells in the renal cortex. That really struck me as going to be a marker of how well with some of these therapies moving forward do we eliminate clones that make this pathologic antibody. And again, drives me back to the idea that I don't think that renal pathology is at a point where they can detect that yet, but moving forward, they very well might be. And using that along with serial changes in biopsies of fibrosis as an even more precise way of gathering effectiveness of therapies. I mean, what we did with proteinuria just made an explosion in IgA, which is great. But we have to admit, at the end of the day, proteinuria is only so useful. So I'm advocating for serial biopsies. I know I do it a lot.
Yeah, agreed, Jim. Harder to make that case in IgA nephropathy than it is in like lupus nephritis trials when people start to go into relapses. But I think I know of at least 1 or 2 trials that are trying to do serial biopsies. Okay, I want to get back to
Barbara. It looks like just because Elaine may need to drop off for another call and asked a question that I think probably we do need to talk about. Elaine asked whether sharing of just placebo data whether it'll be easier. And I think that is a fair discussion to have, but I do want to flag that if all we have is placebo data, we'll be able to answer questions like who's at high risk of progressing, may get better at that. But fundamentally, to be able to understand better the use of and move to a paradigm where we're changing our basis for approval, we're going to need to understand the extent to which the treatment effects on proteinuria, GFR, whatever you have there, are predicting the treatment effect, I guess, in this case, on sort of the 2-year rate of loss of kidney function. So placebo data alone are not likely going to help us move to different surrogate endpoints or validate proteinuria as a surrogate endpoint.
Okay, good. I'm glad you saw Alan's comment. Alan, are you still on, or you might have jumped off already? Okay. So, we all know incentives drive behavior. I'm very curious, and if you don't want to speak up here, then maybe you can email us. Does anyone have ideas of what kind of regulatory incentives would be appealing, especially when you all have to talk to your C-suite, your CEOs to make this investment and explain to them the benefits of data sharing? I think from an academic standpoint, we all understand it, but like Alex mentioned, there are commercial realities. I'm just putting the thought out there. What kind of regulatory incentives do you think could, drive behavior and still make the companies feel like it's safe and it doesn't kind of put anything at risk for them? So if anyone's brave enough to come up with ideas and speak them, great. Otherwise, you can always email us some of your thoughts.
And maybe in that silence, I'm also going to just note, I do think, and I very much want to say thank you to our colleagues from industry who have joined the call today. I know a number of you are developing products in this space. It would very much help us if you could, share your perspectives on some of these issues in terms of what could incentivize you. And that's great. Radko, I don't know if you intended to come off mute, but I saw you came off mute.
Yeah, thanks. Yeah, there is one aspect which I would like to emphasize, and I've been thinking for a while, because do you hear me, by the way?
Yes, we do, Radko.
Okay.
Thank you.
Yeah, so, we have multiple new treatments coming, and we assume they will be successful. They will, especially in combinations, they will slow down the decline in kidney function. So my concern is about new studies, new therapies, new testing, because it will be more and more difficult to put together a control group with some kind of a, decline in kidney function, and to show that new therapy is actually better if we apply all these coming new therapies. So one of discussion points, Barbara, as you mentioned, for company leaderships may be that sharing of data will be necessary actually to construct or to put together control groups, be it active control groups or placebo controlled group, because those patients will be on a new standard of care, and that new standard of care may lead to pretty slow declines in kidney function and potentially to low proteinuria. So I think there will be future challenge here.
Yeah, that's a great point, Rocco. And I mean, you're making me think of that, of the master protocol design, where you do have that common control arm that is shared across the 3 or 4 different products.
So we will have more patients in those control arms because, you know, the potential treatment effect may be smaller for that new drug being tested. So in the future, so that's why it will require bigger or larger control groups, which the data sharing may theoretically may help here.
Okay, great point. So in the chat, we have Michelle commented as one of the potential regulatory incentives would be additional exclusivity. So there's one idea. And I want to move to a comment from Jeanette Lowe. So Jeanette, if you're able to speak, do you want to verbalize this? Okay, I'll go ahead and read your comment. Many countries and regions have strict data privacy and protection laws, which allow study participants to withdraw their data at any time. If the trial data are shared externally, their requests to withdraw won't be able to be met. Will that pose a risk for sponsors? Somebody else commented that, you know, all of this data sharing would have to go into the informed consents. But that is a good question I hadn't thought about. Is this just a matter of maybe, you know, adjusting the informed consent to say, even if you decide to withdraw from the trial, would you still be okay with your data being shared, even though you're no longer in the trial? I don't know if we have anyone from who has thoughts on that. Oh, Patrick, I see your name is your hand is up. I don't know if you're responding to this or you have another thought, but.
No, I had a question for Hito, so maybe I can give a minute for somebody to respond to the thought before I get going. Unless.
Okay. Anyone have experience with this? We're trying to tweak the ICF to accommodate this issue. Again, I'm looking to the sponsors to comment, see if they've tried. Okay, so someone's comment, Laurel, I wonder if the fact that it would be shared in fully anonymized format, that would mitigate that concern. Laurel will remind us that she's not a lawyer, but that is a good point. It would be anonymized data. Okay, so if no one else has a comment, Patrick, I'd love for you to ask your question. Unfortunately, Hito did have to drop off a few minutes ago, but maybe it's still worth posing in case anyone else has thoughts.
So my question is a little bit to follow up on Radko's comments. So it seems to me that to address your concern, Radko, which is perfectly on target, is that we should probably think about future trials to be focused on patients with the highest risk of progression or rapid progression. Yeah. And the question that I had to Hito, but maybe there are others on the call who are much more familiar than I am with data sharing and the granularity of the data that is shared, is does this allow us to imagine a targeted population for clinical trials that are really focused on patients who are most likely to progress rapidly and despite, let's say, the therapies that we have now. But really, the ultimate would be to have precision medicine to try to figure out what drug might target the best, or which patient might fit the best targeted drug, but which would require biomarkers and stuff that we may not have without biosamples. But really, is the granularity of the data that we are sharing allow us to better target patients at high risk for progression?
Patrick, I don't know if you're directing that directly to Radko, but don't you feel that many of the trials are excluding the patients who are really at a high risk because, you know, David, do you think that the criteria should expand it to more of the high risk patients?
Yeah, you know, maybe Jonathan can take this question
Okay.
for us. I mean, so when I look at the graphs that Jonathan is showing, the thought that goes through my mind is, you know, who wants to be contemplating putting patients on a lifetime of therapy for 30 years to prevent end-stage kidney disease 30 years down the road? I think, and it makes me a little bit anxious when we're lowering the not that it's wrong, I'm just thinking, you know, when we're lowering the inclusion criteria to now very low levels of proteinuria to qualify for clinical trials. And I think we all know that there's a huge heterogeneity of rates of progression. So my question is, can we leverage the data sharing that we have to better focus on the patients at very high risk of progression, or differentially design the trial so different trials are targeting different people within the heterogeneity of the disease? Jonathan, I'm sorry to put you on the spot, but I know you've thought about this tremendously. Yeah. So
it is a really important question, and the challenges of the biomarkers we have. I think even though there's been a gram cutoff for many of the trials, people have sneaked in because we have either gram per gram or 24-hour UP of 1 gram. There are a cohort of patients with lower levels of proteinuria that we could work out, you know, what their actual risk is from these clinical trials. I think we're nowhere near having the biomarkers to help us select those patients at the moment. And you're right. The challenge is those lower risk patients, lower immediate risk. We'll need a lot of them, and we need to follow them for a reasonable amount of time before we know whether a drug has an impact on their outcome. But we can't ignore them because they still carry a significant lifetime risk if you're 30. So we need to think how we're going to include them and what the incentives might be for those patients to be included in clinical trials. I know when I've been at the FDA, there's a very strong view that they want to see the impact of the drug across the full range and spectrum of the disease, going down to lower levels of GFR. And it might be that we want to also know the impact at lower levels of proteinuria. I think we can really work through these if we are allowed to see data and look at those data in the whole in terms of across studies, both in the placebo group but also in the treated group. But I don't have an answer for you now, unfortunately. But we cannot ignore these people with lower levels of proteinuria because they are at risk of kidney failure, you know, and they're in their 30s, and that we need to think how we're going to manage these people.
Definitely, Dr. Barrett, you're absolutely right. And there's many of these people who, like for me, I was 23 when I got diagnosed. It's lower levels, and if they're treatment immune like me, other drugs like this of what you guys are all creating is what's going to help differentiate them between being a parent or not. Even though they have low levels of proteinuria, their risk of progression is greater, and wanting to stabilize it a little bit more as well so that way they can have that chance of having a family, because that's something that is very important to a lot of these patients, is getting their their disease stabilized to where they can move on to other things. They might not be 2-3 grams, but they might be teetering on like 700, 900 in that area. And many of us already know we're going to be on drugs lifelong, so it's really not a big, it's not a a deal breaker for that. So definitely targeting those that are lower and not already damaging their kidneys a little bit more than the rest of us. But I think it's definitely an ideal population to do these studies on.
And it looks like, we are running out of time for this. I don't know, Barbara or Uptal or John, if you're going to close this out.
Yeah.
I do want to thank everyone. I think if anything, this highlights some of what we're up against in terms of actually maybe moving forward with sharing of data.
Can I just, so to that, just to say this, so lots of people on the call, I recognize your names, and I recognize we have had confidential conversations around this area to a great extent. And I completely understand why you might not want to verbalize that in an open forum. But I think it's fair to say, Barbara, that anything that's sent to the KHI group, i.e. yourself, it will be treated confidentially. So please, can you let us know what's on your mind? And I think it's fair to say, Barbara, that the KHI are very interested in reinvigorating the work group to rethink about endpoints in IgA nephropathy if there is enough data to make it worthwhile. But I would strongly ask all the people on the call from industry to, in confidence, let Barbara know what is on your mind and what are the questions that perhaps you weren't prepared to ask necessarily in an open meeting, because we desperately need to know there is an unmet need from you guys to justify going through the whole process of setting up another work group.
Yes, thank you, John. KHI has always been about creating a safe space. And I'll even say, don't email me. You can email, I think it's KHI@asn.org. Maybe Alicia or Abby, you can put it in the chat and we can send it to everyone. And if it goes to the KHI central staff, none of us can even know who, and they can create a spreadsheet of the questions without knowing who's asking. Before we close, I do want to point out something we didn't get to, and maybe again, you can email us. There have got to be other therapeutic areas where this has been done and there have been lessons learned. So if any of you in industry, I know you don't always do kidney disease, if you have experience in other therapeutic areas, genetic diseases, other registries, and you'd like to share that, please, again, email that to us. Next time, we're going to make this an hour and a half, Abdul. But okay, I'll just say a quick goodbye. And Abdul, thank you, everyone, for joining. Abdul, I'll give you the last words.
Yeah, thank you, Barb. Thanks, everyone, for participating in the active conversation here. Hopefully, this is the beginning of a longer dialogue. Again, encourage you to reach out to the KHI email there, follow up, and continue the conversations. We can't move this field forward without data. And we've come a long way in 10 years. We still have a long way to go for our patients. So thank you again for your engagement. And thanks to the KHI ASN team for hosting this. Have a great day.